Good morning, good afternoon, or good evening, wherever you are in the world today, and I thank you for joining us as we kick off our live Q&A following on the tail of the Small Talk webcast. This was the first time that we did something like this through Beckman Coulter, and working with these incredibly talented scientists, Dr. Alfonso Blanco and John Tiggs. We're happy to have you, and we're really excited to get some feedback and to hear what you have to say and what questions you have. Thank you in advance to everyone that has submitted questions. And without too much further ado, my name is Matthew Goff, and I am the marketing manager in North America for Flow Cytometry. I've been in the background hosting these fine scientists throughout the process, and so I'm happy to have them with us one more time to answer your questions. We do have a few submitted questions that came in via email and a couple of other channels prior to the start of this event. So we'll kick off the chat with a few of those questions, and then we will go to any of the questions that are asked in the Q&A box of this session. So without further ado, I will start the question and answer session with the following. Do you, you'll forgive me, I don't have an attribution for this, but do you suggest using logarithmic or linear settings and why? We usually look at things in log, but are working with another group who used linear, which requires voltage to see any events. And so to Alfonso and to John, I kick this first question to you. Linear settings or logarithmic settings in terms of the data visualization, and why?
You want to start and then I follow, John?
I mean, we can. I know you were getting together something to actually show it in
yeah.
Realistic fashion, but I can't imagine being able to see it all that well on a linear. I would say on a linear scale, you'd cut it off on a higher end, probably 500-ish. You wouldn't get low enough to get into the hundreds on a linear scale. And I would think if you have to put the gains or the voltages that high, you may actually be generating false information just coming from the opposite end instead of low end noise that you talk about in these instances, coming from the higher side down. Because I've had it in linear, but like I said, I think I cut off somewhere around 500 that I'm able to see. Do you have the actual example?
Yep. Just 1 second. Let me share the screen for a second. Cytoflex, where are you? Here. Right. Can you see that now here, right? So this is forward scatter area. I can change this to high because we will be working with EVs. So I'll change for forward scattering height and side scattering height. And again, you can do this with any other instrument. This is what we are working with gains. In other instruments, you're working with voltages. But you can see that the data is quite compressed there. You don't see it. Okay, we can make the zoom, and you can look at that. And now you can see one population there. And this is more or less what you will see. And it's hard to define what is the other. Now, you're missing a lot of data down there. If we play with log, let's change this to logarithmic scale. You can clearly see the distribution of these ones. We are working here from 10 to the 3 to 10 to the 4, and you can see if we go down and up again, you can see that we get data from 10 to the 3 to 10 to the 5. And I go to Megamix, and you can see here all those data. If I convert this back in again into linear, you can see how difficult it will be to try to define those populations. Even if you play with the voltages, we will play here with the zoom. It's quite hard to get all the distribution in just one vision. So usually what I do is I compare these log linear conversion into zoom in, zoom out. If I want to see everything with a perspective, I want to go back and see everything. I want to see everything with a big perspective, and then I will be working with logarithmic. If I want to see something with more detail, then I will change to linear scale. And this is something similar to comparing why we are working with logarithmic scale and linear scale when you're doing cell cycle analysis. If you're looking at big distributions like GFP expression or CD4 or CD8 expression, you want to work with logarithmic scale. If you want to see the difference between G0, S phase, G2, those data are really compressed. And then is when you will have to work in linear scale. Probably the reason for your supervisors to work in linear is because they have been looking at different trainings where they say linear scale for cell cycle and for forward scatter and side scatter.
Yeah.
You're working with the scatters, you have to work with linear. Yes, but no. Yes, if you're working with cell lines, if you have very well defined populations. Yes, if you're working with populations that they have very small difference in sizes or density and granularity. And this is not the case on EVs. You can see events that they're going from 80 nanometers all the way up to 2 microns or 3 microns. So it's a big difference on sizes. It's a big difference on densities. For EVs, I will definitely recommend you to work in logarithmic.
Perfect. Thank you guys for such a detailed answer. And of course, this is being recorded, so we'll be able to capture your walkthrough for that, for those people that are interested in seeing the difference in the settings. The next question, I'm going to take a little bit of creative license in interpreting, but can you all describe what steps you would or would not use in the fixation of EVs, the preservation of EVs for analysis after the staining process or after the extraction process? The specific question related to formaldehyde or paraformaldehyde, but I think it's a good enough topic that we can sort of peel back and dive into a little bit.
I've thought about this one more and tried to look at it from who's actually done it. I don't think people fix them simply because, and I'm going to say in my hands, they tend to last without having to do anything to them, like cells you have to fix and whatnot. They start dying. I'm not sure in these, no one's really done viability studies just because it's difficult enough to work with them to then start worrying about, do they like cells, apoptosis or necrosis or anything like that. So the fixing hasn't, I guess to even bring it down to a lower level, we've left these things on a desk for weeks and have gotten similar results just running them. And some of it was simply us wanting to do that just to see, can we keep them? And we've had no problems, even with them being labeled, because most of them are membrane dyes or something like that. So you don't start really losing fluorescence intensity. So do you need to fix them? I don't believe so. I haven't seen anyone who does it. And I think having this chat with Alfonso before, and you can add to it, it may actually cause some changes that you wouldn't want. Could you do it? Sure. And it goes back to how the talk went with, if you do it, then just keep doing it. But I don't think people actually do it. I did a quick literature search and couldn't find it.
John, I think the question is kind of because people is looking that for SEM or TM, they are fixing them on PFA.
Yeah.
And if they do it for microscopy, then we have to do it as well for flow. And because we are used to fix them, it's great. On the other side, it's a long process and a painful process to prepare all the samples, and it will take me all day. And if I can prepare them today, can I run them tomorrow? For that, I have to fix them. It makes sense. The downside is if you fix them, I guess you'll have some shrinkage, and that's not what we want because they are small enough. There will be changes as well on the membrane and the distribution of them. So that we will lose some capability to try to differentiate them. They might have problems also with the cargo If you suddenly fix them, they might lose some of the cargo. And again, we might be losing information of what the real content has. So I wouldn't recommend them to fix. If they do it, please let me know how and it works. And again, as you said, whatever you do, just keep doing it in order to compare them.
Could I ask just a one layer deeper question on this one?
Yep.
And that is, are there any preservation techniques that you have to take into consideration for these? Could it be like for temperature? Do you have to take into account freeze fracture? Any sort of chemistry concerns in terms of, you all mentioned membrane dyes, and there's a pretty well documented enigma with PI where it'll leach back out of the cells. Like if you sort them into PI cells, if you were to sort them into a buffer not containing PI, right, you can lose some of the signal because it'll come back out. Are there any concerns like that when it comes to these sort of structures, or is it less so just because they're non-nucleated and so they don't have an active ability to effuse or infuse?
I believe what you said, yes, is true, in the sense that I have not seen that this has become an issue of things leaching out, or any of that kind of problem. Freezing, obviously people do that. I mean, they normally spin down, freeze them, and then it's the same type process in the freeze thaw. None of it wants to be rapid. And I think we mentioned it, problematic is in the first processes. Like if you freeze or chill out the blood, you'll have issues there. But people take the samples and they'll, because you get patient samples, and in order to get enough and to have those kind of cohorts, a lot of people take it and then freeze them down and then do all kinds of work afterwards. I've seen that many times. We deal with a lot of fresh stuff. So we're not worried too much about these things. Like we haven't had to overly think about it because everything's done pretty much in a rapid format. And the most I think we've left them is maybe overnight.
Mm-hmm. Yep. So one of the things you said is that they will not break down or die. They break down. And this is why we have to be careful. And in fact, one of the things you can see if you're sorting them, if you reanalyze some of the populations, especially the bigger populations of EVs, is that they break down. Looks like the sorting hasn't worked, but the sorter has done the job. It's just basically they break down, and you can see small populations showing up where they shouldn't be. So then again, you have to be careful, and whatever you do, keep doing it the same way. How many parts you will have afterwards, after they break down? No idea. So you have to be careful not to damage them.
Yeah, but I think that's more in the processing.
Yeah, it's more processing. Yeah.
Once you have them and they just sit around, they seem to be pretty robust.
Yeah.
They're little tanks. Like you said, Matt, they don't have a bunch of things that may cause, attack, and you have normal things that undergo attack by other stuff, and that makes them die. It doesn't seem like many things attack them, especially if you're just sitting in a tube. There's nothing else there. Mm-hmm. So, but like you said, if you're processing all of them, and same point for TEM, cryo-EM, they tend to get fixed and squashed and all these other things, and that's why a lot of it was like, well, are we sure of the size? Because you're actually flattening them out instead of keeping their shape. So. I have not witnessed this. Sure, it can happen, but I mean, that's something that would be interesting for someone to take a look at is, long-term survival. So
let me see if I can jump on some of these questions that are coming in live. We'll just kind of work them in order along with some of the submitted questions. So from Vladimir Christoph, is there a way to ensure ascertain that EVs being analyzed are single particles and not clumps of two or more?
Go back to your Edwin van der Poel swarming paper. So serial dilution. It's probably the only perfect method in doing that you don't have clumping. You just sit there and obviously do your serial dilutions, and you shouldn't have major changes in your population. In flow, it's very easy to see because it starts off and it just keeps flying up the scale, and the speed goes out of hand. On the Cytoflex, and there may be some others that do it. Obviously, your sorters, the abort rate gets out of hand also. So it's just an overwhelming of the system that you see. So there's the single EV has become a topic that has been kind of covered lately between the swarming, looking at things. I know Alfonso uses the same technique as regular analysis where you do doublet discrimination.
Mm-hmm.
So you use the different scatters in order to.
Have you read my mind?
Yeah.
Preparing one for them.
Yeah, he's preparing it. So I'm in his head, even though we're not even near each other. So we are extremely socially distanced. But you can actually see it live and in person. If you haven't read the Van der Poel paper, I suggest you do, because a lot of things that are based on the, kind of MySev rules start there, getting and making sure that you're not just looking at clusters of things, whether it be clusters of EVs or clusters of dyes. So dilute.
Yeah. So
Okay.
I was preparing something in the meantime. I will share again the screen. So usually we use area and height, and here you have, these are the apogee beads. You can see violet side scatter on height, violet side scatter in area, and you can clearly see kind of the clear diagonal, but also you can see here parallel population showing up. As long as you started to dilute, you will see that population that you can see there disappearing. So it's a good way of checking that you don't have. If you see there, the number of aborts in this case is 35%, which is exactly what John was saying. As long as you decrease that number of aborts, you will start to see that this is decreasing. So if I go to another sample, you still see that population growing up. You have less aborts, but that's the reason because we have removed all of this with the triggering. So be careful with the number of aborts. And it's good to have always this double check that you don't have these aggregates. Especially in violet, I find out it's quite simple to see these aggregates. You can use any combination of scatters. So you can see side scatter area and side scatter height. And you can see again, this is in linear scale, back to the first question, and logarithmic scale for both of them. And again, you can see those aggregates going in parallel. The advantage here is we can start to move things down, put them back into scale, and you can see where we have the aggregates. You can see we have less aggregates when we look at the blue side scatter compared to when we use the violet side scatter. So my preference is always start with violet side scatter for removing aggregates.
Yeah, the other thing too is if you're not even looking at something like this, the other point is if you're just looking at normal logs or whatever, or even fluorescence, your intensity shouldn't change as you dilute. So as you increase the dilution, yeah, you'll get less events, that's obvious, but your intensity should not change.
Mm-hmm.
And so in our hands, we find somewhere around the 1 to 1,000 tends to be a good dilution when we're playing with viruses or algae in this case, or plasma. So, and that's a good starting point. I've seen papers where they do dilutions from 1 to 10 to 1 to 1 million. So.
Mm-hmm.
Okay, very good. If we can go to the next question. Renee has asked us, do we prefer triggering on size or, and I think Renee probably means scatter, or do we prefer triggering on fluorescence?
It depends on your question. I think we covered this discussion on session number 5. And for me, I prefer to look at size triggering rather than fluorescence. And The reason for this is because I want to see everything first, and then I will focus. I could be losing some information if I'm triggering just in the fluorescence because I want this exact population showing it. And you're looking at monocytes, and suddenly the ones that are making the big difference could be platelets, or could be some macrophage doing something absolutely strong, and you're missing it because your boss told you to focus into monocytes. Why? Because I'm a specialist in monocytes. So I will first started to look at size triggering, and then in fluorescence. And again, we had some examples where we started to work at triggering on fluorescence because they were looking at platelet-derived EVs, and suddenly what you're looking is basically at platelets, not EVs. John?
No, I agree. I've always kind of been in the world of scatter. That's how I started, and tried to build things around that. So, my original build of an instrument was kind of a Frankenstein, and it was all based on scatter, right? It was using the forward scatter and really enhancing that. And so then we were looking, and a lot of things no one labeled. I guess fluorescence is each, how do I easily put this quickly? Each has its benefit now, and each has gotten it kind of a pickup over the years. So using scatter properties has really, people are starting to do it more and more as the instruments have come along to do it. And so software has been written. You have again, Edwin Vanderpoel, mentioned him. He has his Rosetta and me theory calculations. Joshua Welsh at the NIH has put together FCM Pass, a free software that will characterize your scatter properties and your fluorescent properties. So like I said, as these things get better, you're able to do both. We've started doing more fluorescence just because markers got a little better, such as using lipid dyes or like a cell tracker, people putting actual GFP on things and stuff like that. So you don't feel like you're just, like Alfonso said so eloquently, if you use the fluorescence, you almost feel like you're targeting one specific thing and one specific thing only. So it's whatever labels, that's all you look at. And whereas there can be information outside of that, just in the scatter world, that you're missing. Mm-hmm. So, and then as you start multiple colors, it becomes, which one do I trigger?
Yeah.
So can I ask you guys to go a layer deeper, since you're both fans of the scatter, and I'll try not to use the terminology embrace the noise, but my question is this: there are a lot of different flow cytometers out in the market that have the capability, with proper tuning and data manipulation and settings, to show you EVs. But based on scatter, there's several different ways to do scatter, right? There's forward scatter, there's enhanced small angle forward scatter, there's back scatter, there's virtual side scatter, there's violet side scatter. Could you guys maybe dive into just a little bit of the differences in the scatter, and maybe if there's one that you particularly are fond of for a particular application, or when you would seek something out versus something else? And I bring this up specifically because I know that of course the Cytoflex offers violet side scatter, and then the MoFlow offers a different sort of, or that MoFlow Astrios offers a slightly different scatter. And so I think it's worth talking about both, since both have been implicated in EV research.
John, you start.
Okay, so I've played with many different scatters. So like you said, we'll stick it to kind of the Cytoflex, the MoFlow, my major players. I have some other things, and they're violet side scatter based also. Violet side scatter in the sense that most of these things were based off of me theory. So the lower the wavelength, divide basically by half, and that should be your lower end of detection. It is slightly lower than, if you did a 405, it should be 200-ish, but it goes down even further. And it's more than, now you can use focusing lenses, right? And get things perfect. The same aspect works if you're doing forward scatter on an Astrios, because now you're focusing again to a PMT. down into a pinhole, and you're kind of focusing all the different light. And we think of it as like the Young's double slit. You're taking the light that kind of scatters out through a blocker bar, and you're getting each through the ends there. So the thicker the blocker bar, you're getting the direct light out, so the noise goes down, and collecting all the higher incident light, the bigger angle. And so that's why it works. If you're using blue light, I've tried them all on the Astro. So they have the 7 laser. Obviously, you can't do it with UV, but from violet, blue, greens, yellow, greens, reds, they all work well. And in theory, infrared should even be better because, in sense, yes, it's a higher wavelength, but once you get that high, things kind of change. You lose the noise, you get less speckling off the laser, so you get a higher dynamic range. And we've seen in the far reds some indications of these. You get some advantages to it. So diving deeper into that is all of them kind of work. I think what it comes down to, like you said, you can use polarization. I mean, DLSs use backscattering and side scatterings and forward scatterings and take the 4 and get you a picture. But I think it becomes an ease, for one, is how do we get a base unit and do something easy to make it do something else, instead of trying to kind of be specific to the one task, which, don't get me wrong, I want something that is specific to the one task because that's what we do. But if you're just a regular lab and you're like, oh, I have only one person who's starting to dabble in the exosome world, it just makes it easy to convert the machine to do that analysis. And that's why fluorescence became such a hot way to do it, is because all the machines could do it. And so you didn't have to go out and spend more money to get it done. So I think that's what it comes down to, right? Is what you have available in your hands. I find that I've done the comparisons, and the violet scatter on one instrument works just as well as violet scatter on another instrument, which works just as well as, say, a regular side scatter off of another instrument. And this becomes because you're not just talking about the laser lines anymore, you're talking about all the optics that are associated with it. So by putting aspheric lenses, by using different blocking bars, by using different detectors. So that's my 2 cents.
There's a few years ago, there used to be the ortho. It was a flow cytometer that has an orthogonal side scatter as well. Talking with engineers, because I haven't used it, so I was just chatting with people, and they said that they were basically removing that scatter because it was not giving any extra information. So it's the same than side scatter. So I'm just waiting for a company building up something with forward scatter, side scatter, orthogonal side scatter to see what else is coming out. Because when you think on the scatter and how these particles are moving and flipping around, the way that they are scattering could be absolutely different, especially if you're thinking not just on the EVs. I'm thinking now on the nanostructures, or when I was talking about the bacteria or the phytoplankton, the shape of this could definitely change completely the ortho and the side scatter light. So it could be a good way to move forward, especially for the people in physics, to have a look at the orthogonal against the side scatter in multiple lasers.
Excellent. Thank you guys for tackling that one. Let's take a look at our questions again. We've got a few more that have come in. The next one is specifically about the Cytoflex. So if you guys will allow me, I'll take that one since it's a spec question. What is the limit of detection on EVs on the Cytoflex? The limit of detection on the Cytoflex for small particles is tested using polystyrene particles, and those particles have a refractive index that is not completely like cells. And so we know that in scatter, we can resolve 80 nanometer polystyrene particles on a stock Cytoflex without any sort of adulteration or special order or changing. That said, that is the 100% compliant qRNA approved brochure printed answer. We do know that when combining scatter and fluorescence, some researchers have been able to use membrane dyes, and membrane dyes combined with scatter and surface marker immunofluorescence, for lack of a better term, and they've been able to get down as low as and even just under 60 nanometers based upon correlating SEM data. Past that, you were in a very, very gray territory, and just like anything else, I would say it's true for any instrument that the closer you push it to the edge of its resolution, the more argument you're going to get from the community as a whole as to what you did or did not actually accomplish. So if you're planning on chasing the edge, my recommendation to you is to verify and correlate your data to other methods that that will show you that what you're saying is true, and that's probably the best practice. Okay, I think that
Agree.
is that a good enough answer, gentlemen?
Good answer.
Okay, thanks. Let me see. We have another question. I'm going to extrapolate a question and see if I can ask it in a way that's general and specific enough at the same time, but it relates to sample mixing. And the question is this: whether you use an up-down pipetting mechanism or a stir stick type mechanism, you're liable to introduce bubbles during a vortexing step. And I believe it was in episode 3 when Alfonso was kind enough to show us the Irish method or the Spanish method. And and we want to avoid, obviously, the introduction of air bubbles, as those those trigger the count, right? And they look like events. It's data that we have to deal with that could be very similar to our EVs in terms of the signal. So the question is there a way, what do you all recommend for, especially I would say for plate-based assays, what do you recommend as a best practice for minimizing the introduction of air bubbles into the sample? Maybe we can talk about air bubbles in a more general sense and sort of what their implications are and how to avoid them.
How to remove them from the analysis. Oof, good question. Thank you. So I guess that one of the things you can do is basically look at the pattern that they draw in your PBS alone and see how they look like and where they are falling. Because more or less you will see after a while that they are going in one particular direction, and see if your subpopulations are affected or not by those air bubbles. When you're playing with different scatters and different buffers, you can see sometimes when you place scatter against scatter, you can see kind of a shape like this. And that shape is basically the buffer and maybe some air bubbles and some debris. So if I see that shape, I definitely recognize that it's not something that I have to take in consideration. I know there's something I have to discard, and then focus into these other populations that are showing up. I will try to find something like that. I mean, John might answer something else, and I show you that shape that is quite characteristic. And I will try to find that way. The reason why I found out, in fact, it was not me. It was a user that we have here, Carl Egan. He was basically running the sample 3 times because we want replicates, and he also ran the sample at the beginning of the experiment and the same sample at the end of the experiment to see any changes, as John was saying, just to see how robust my sample is in terms of labeling, for how long I can be running here in the flow facility. My samples could be 1 hour or 10 hours. So when he was running 3 times consecutive the sample, he find out that suddenly he has more and more and more events, and always in the same area was the population growing up. And that's the way I found out they were air bubbles, and we decided to turn off the steering. But John, I'll pass you the question. I will try to find this file.
Okay.
And then I'll come back.
Yeah, I mean, So I understand on the Cytoflex, and I was reading the side so I could get a better idea. On the plate mode, you can't turn it off. You can turn it very low. Like I've done it where it goes in and it just goes boop, and it stops. So not as much. Now, what I've seen in a little bit, it is hard to duplicate unless you start getting to the top. So in tubes, it's a lot easier depending on volume. Like I said, we're diluting, so we get a lot of volume. As the probe goes down to the bottom, the bubbles aren't as prevalent because bubbles go to the top. Plate's a little more difficult simply because the volume, surface area, and you may get them there. But I don't think that they cause too much issue if the probe, as the probe goes down, you shouldn't be taking them up because you can't get around it at all. You'd have to let it sit, then it goes back to then how do you mix them? And so you end up fighting. And I think flow cytometry is a little more forgiving of a method with bubbles on this type thing than a lot of the others. I know microscopy loves bubbles because simply in order to look at these under certain things, one of our microscope experts likes to put a bubble in so he can use the bubble to find the edges and then come in to look at the EV populations because they're so small. But I think in a sense, the pressure, right? A lot of times the pressure that's put onto the actual sample gets rid of a lot of the bubbles. Because I know we've talked about this, right? I guess I'm talking to Alfonso and Matt, but we've had the conversation with, it was the machine that Beckman Coulter actually has, and I was watching videos on it, and they add pressure to the actual sample in order to get rid of the bubbles. And I think I said in one of the series, it was the crazy guy running basically like salsa water or something like that. And he added pressure, and the added pressure would cause the bubbles to go away. In some aspect, I think that the tube pressure that's put there throughout gets rid of a lot of those bubbles too, because you can't completely, especially in the plate, stop it from doing its task.
It would seem to me too that the use of a 96 deep well, if the depth of the tube or the vessel is what helps mitigate and separate the air bubbles from the draw point, on the sift for the sample, the top of the sample being further away from the input would help with the bubble mitigation. So maybe 96 deep wells are something worth exploring for people that are encountering this issue.
Because in order to turn it off, I think you'd have to actually get into like the service side of things.
It was a specific question from someone I believe that has a Cytoflex, so we're gonna take that one offline. But more to the point, it seems as though best practice is, regardless of the instrument, that turning off the automatic mixing will help mitigate the creation of bubbles.
I'm afraid I cannot find the file.
He's fired.
Next.
It's okay. While you stall. So we have 20 minutes left, and we've got 2 more questions that we want to get to. I think these are gonna take a minute to answer. We asked about fixation earlier. The other question is, let me see. I'm going to extract this and ask it again in my own way. Is there a recommended method for permeabilizing an EV and rendering it intact and doing I hate to say intracellular, but intra-EV, intravesical staining, if you will? Has that happened in your hands, or do you know of any other prominent papers that have a recommended method for this?
Not to my top of my head.
Okay. Oh no, it's published. So one thing.
Sorry.
One thing we've actually used for doing our molecular beacons. So we want to get the molecular beacons in so they can tag themselves to the microRNA, as an example. And what they've attached them to was a cell-penetrating peptide. So this allows for this to get directly in, perforate the EVs and get inside. I also know with something like a CFSE that was done by Jennifer Jones, she has, one of her papers of labeling that comes in. the CFSE does get into these cells because you can light it up. You need an enzymatic reaction and then it happens. Some of these you can actually add certain things. I think what you can actually do is calcium fluxes cause channels.
Mhm.
And the other thing people have done is actually use the cells in culture, say it's a tumor cell or something like that, labeled that, and when they bleb off, some of that same reaction happens to their friends, their little friends that they send off, because a lot of these processes with the exosomes take place in the multivesicular bodies and then they're ejected. So I know doing like GFP proteins, they get in. I'm trying to think of what else you want to label inside, because technically the only thing in the cargo that you'd be going after is like some sort of RNA.
But all of these without fixation or permeabilization, right?
Yeah, I don't know how they'd react to an actual permeabilization technique, if it would even do anything.
So
what
so
I
you
always
so you
say
end up
is
using the
try.
same tricks.
Yeah.
Go ahead. Sorry, Alfonso.
No, what I'm saying is, as always, we recommend is try, play, and let us know, because it's not there doesn't mean that you cannot do it. But I will play with controls all the time, and again, I will try to look at the populations before you fix them up and after you fix them, and see how much they have changed.
Yeah. I mean, I get it, but I guess some of that question would be, what's the target? Like, what are you trying to permeabilize to get to? Because if it's just the RNA, then yeah, a CPP is probably going to work. Other things just naturally intercalate. Cells will take them in just because of changes in their environment. So like I said, if you're causing changes in the calcium, you may be able to open things up and place things in. But yeah, I'm not sure of people who have actually gone looking for anything else, because it's just not, like DNA is not there. You're not going to be worried about trying to get nuclei, like you said, Matt. So, and a lot of those dyes anyways, if you could target DNA, I mean, there's some that may say certain ones do carry DNA or mitochondria. You can find those papers. Then those type dyes are going to pop themselves through anyways.
Yeah, for
Perfect.
dyes, yes.
Yeah.
Fixing to introduce antibodies, I don't think so.
Okay,
But
very good.
it's a question of trying.
Trial and error.
Yeah.
Let's go to another live question then. Do you have any recommendations for do you have any recommendations for the best ways to titrate your antibodies for EV research?
Oh, thank you. The first thing you have to do is make sure that you have the right concentration. So I will start first with the concentration of your particles. Make sure that you have your particles completely concentrated. And then from there, you will start to work with the antibody or your dye, and you will make the dilution of your dye. I will work first with the dye alone. And again, remember what we were saying, if you're working with antibodies, you need to remove any kind of clamping on the antibodies. Make sure that your dyes or your antibodies are not rising up the levels of the background or noise. And then from there, you start to look if your signal is going higher and higher. Again, I think it's controls.
Yeah. Antibodies give themselves a whole different array of problems, right? So a lot of the ones that you try to label with now are coming with kits. So if you're talking about tetraspanins, you can get them in kits in that, cookbook kind of method. If you're using something that may be a little bit different, then yeah, it's doing the titration curve. And you'll find that some work better than others. We may have mentioned it, we may have not, I can't remember, but it's clone specific also. So some clones work exceptionally well. Other clones of that same thing are horrible. You won't get any labeling. So this is very, some people, do titration curves for everything. Some people don't even know what a titration is. It's like a nasty word. sometimes in my flow core, you tell people, oh, they're like, oh, why didn't it work? And you say, well, did you do a titration? And they go, what? So here you don't really have that option. You kind of have to go. Someone once joked, hey, start with Avogadro's number and work backwards. And it has a sense of truth to it, which is sad. So it is trial and error definitely for this type thing. And a lot of the companies will work with you. I know people have used a lot of like the things from BioLegend and stuff like that to use those. I thought about, what was it, the reaffinity type antibodies, since you asked about antibodies, simply because of the way they're structured. And I know others sell similar ones to them now. And that's what popped in my head. And supposedly they don't have background problems in aggregation and whatnot. So be interested to see how something like that worked. We don't do enough specific labeling. We do red blood cells, so we've used GlyA, and then if you're using an Exon-5 for phosphatidylserine, those are pretty well documented. So you might not have to do complete titration, just go to a paper, find out the concentrations from there, and then, do your own little titration just in that wheelhouse.
Perfect. Thank you so much. The next question, I think, goes back to episodes 1 and episodes 2, where we had the robust discussion about isolation techniques. The question specifically is related to the Cytoflex, but I think as a baseline, we've established that focusing on sort of, EV interrogation through flow cytometry as a science, and less focused on the Cytoflex. Is there any, because I don't think that it changes based on the instrument that you're going to use, is there any isolation method for exosomes that you think is proven to work better for, I'm going to say, for flow cytometry? I know that we talked about them in general terms in some of the previous episodes, but I didn't know if, and I assume that you all have had access to either, column or ultra, the variety of different methods that exist, bead-based capture. Do you all have a preference for what tends to give you the best particles post extraction or isolation?
No, to be honest, I think it's more the sample and what they're looking for, and what makes a huge difference is the user. So that makes a huge difference, and you can feel it. You can see which ones are the good samples and the ones that you will trust and the ones that will not trust, and I think it's more the user rather than the technique.
Are there any suggestions, Alfonso, if I could get you to expand on that? Any sort of bad habits that you've seen users do, or mistakes that have been perpetuated that affect the quality, independent of the method?
Yeah, well, the common mistake is doing what my other colleague is doing. One is working with one kind of samples, and the other ones are working with completely different samples, and they haven't checked any literature. And just because the other one is a good guy, I will follow what the other one is doing, rather than trying to optimize their own method from the beginning. They don't want to waste their time, and they don't want to spend time in the optimization process. And that's where you will see mostly of the problems. So I will start always with optimization from every single protocol, and I will never trust anyone in my lab.
And so as a summary, then you would say that if you're getting it from, say, urine sample or RBCs or platelets, all of those are going to have specific optimization methods that need to be verified in your hands.
Yeah.
Okay.
I think we were even mentioning when you're working with cell lines, and suddenly you look at one of the papers and you see, well, they're spinning this at this speed with this temperature. You go to your own centrifuge, and suddenly you see you don't get the same results. That's because the arm is slightly bigger, because this one is spinning a bit faster than the other. So all the small difference, you have to optimize them, and that makes a big difference.
I guess I'll also, since that's a very broad question, I guess I can comment. There are many position papers on this that you can see that balance the, the major factors, effectiveness, cost, maintenance. So, ultracentrifugation, gold standard, right? People do it. But there's a lot of problems to that I have read and seen in these kind of position papers where, like Alfonso said, not everybody's taking into consideration how they should do it. It's just a generic, and you can lose a lot of stuff. You can pellet out the wrong things. Some people don't save the supernatants. So you have these kind of errors, which then allow for other things to start making their presence known. So a lot of people now have moved to kind of size exclusion chromatography, just using the columns because it's very easy. It is kind of cost effective, and you're taking every fraction. So the chances of losing something becomes less. And I mean, I'll say it again, time, right? It becomes a more of a time-friendly thing, whereas you have to basically overnight in a lot of these ultracentrifugations. There's such a long time for it. I've seen a lot of like immunoprecipitations and density gradients and, like you said, capture different ways without giving away too much information that other people are working with. But there's some really good methods coming out that are trying to isolate and get the most back. With any assay, right? There's going to be loss. It's just, what are you willing to accept? And how detrimental to your project is that loss? If you're trying to kind of see, oh, okay, I've got a disease state and I want to see this patient's reaction to something. If you can't get kind of consistent numbers, if you're worried too much about, well, I lost a bunch, how do I normalize it? Then yeah, you got to look at how you do it. The other thing is just maintenance, right? We said maintenance. Some of these things, like an ultracentrifuge, you've got to make sure that they're in tip-top condition, right? You have to have them maintained. You can't just sit there and expect, okay, I've got the rotors, I've done this, and I'm using it for the last 10 years, and I don't have to have an engineer come out to check anything. So it's like, that becomes more of a high-end piece of equipment that needs more tender love and care, just like your cytometer or your sorter. You have to have people taking care of it, have people making sure that it's performing the same each and every time. A lot of these other methods are based, obviously the columns. I mean, they're based on gravity for the most part, and take it in, elute the fractions, and it goes. So I guess it just depends on if you have, a lot of people don't have access to an ultracentrifuge. We have a lot of people come to us to, ask, hey, do you know someone who has an ultracentrifuge? And then on top of that, can I use it? Hey, we can bring it right to this topic right now. During this new normal with COVID-19, a lot of labs are not open to other people coming in and out. So when you were using someone else's ultracentrifuge for your experiment, we are now dealing with this of people going, well, I can't get into that lab anymore, so I can't do this. Mm-hmm. It's like, well, sorry, but that's, kind of how it is. They have the equipment and they don't want you in because numbers have to be what they are, amount of people. So again, who knew this situation would happen, but there's an interesting aside to it, right? That now you don't have the access to common equipment that you were used to.
Mm-hmm. Correct.
I think we joked in the outtakes during the production of the episodes that an ultracentrifuge is akin to a chainsaw. Just because somebody has one doesn't mean they're going to let you borrow it.
Yeah.
And so the possibility for damage and destruction is not insignificant or trivial. So I'm going to use your answer here to answer the next question that came in live from Jessica, and that's, is there a generally recommended high-speed centrifugation speed and time to isolate EVs? And if I'm reading the tea leaves from you both, you would probably say it really does just depend on the sample, the volume, the rotors that you have available, what you're willing to lose. But is there a general, from what you've seen, is it normally a 1 hour process, a 6 hour process, a 12, 24 hour process? What do you all see most commonly?
I think the last step in those is somewhere, it's like up into those like 10 hour, right? At 100,000 G or something like that. Mm-hmm. So I know the speed is like 100,000 G, and then, you have this time frame. Obviously that time frame, it's long, we'll say it's long. how long, we're not talking an hour, how long is dependent on the sample, because there is a fine line between getting that exosome fraction and just slamming everybody to the bottom of the actual tube.
Mhm.
So that's, you know, that's, people have spent a long time figuring that out. So there is, you know, the whole chart that breaks down each step, what you expect to get off from those supernatants and how long you should do it. So again, if you're just starting, there's been people for like the last, you know, 8 to 10 years who have at least given you a stepping stone where you can start instead of starting from nothing and trying to figure it out.
Mhm. Yeah, but that's the beginning. You have to find out as well.
I mean, the good,
Is
obviously you don't have to stare at it for, you know, all that time, but, you know, it's still, you're not going to have your sample, you're not going to get a sample from somebody and have it ready in, you know, in the next 15 minutes to run. So.
is there a particular resource or chart that you all tend to reference or point people to when you're asked this question for settings, or do you refer them to manufacturers' recommendations? How do you normally approach this question when your users ask it of you?
There's some, okay, so there's some really good educational sessions. I know some of them were running, might still be running for free from one of them that I kind of referenced to is from Coursera. And that was a course that was put through, I think it's University of California. And there's a lot of good information from, it's Ken Whitner, messing up his name. I'm drawing a blank and I don't know why. You know, and there are steps in there that go through the processes of centrifugation and things. So that's a good one. I believe there's some that are associated with CytoU. So that I believe you can get onto the what's called the ERCC. That's a consortium for RNA, extracellular RNA consortium, and they have protocols there for isolation. And then PubMed has a ton of people doing just position papers of, you know, what works, what doesn't. Or everything kind of works, but they do these compare and contrast. That paper you sent me on the bronchial lavage, the BALF, it actually has a bunch of that comparison of methods.
Excellent, and I will try to get those resources commingled and send them out as a follow-up for the people that were able to attend.
So I don't have to find it.
This
Alfonso is the librarian with
No.
his many folders of resources. I think we have time for one more quick question, and this one it's pretty specific, so I'm just going to read it straight from the screen. What are some markers that you would suggest for BALF, and what are your thoughts on using D7777 as a membrane marker for BALF? And it might help if you give a quick introduction to BALF and then go from there.
So we were just mentioning it because we just got this question and had no idea. We are just very bright and know everything about anything. So we got the question last night. So Alfonso had looked up a paper. I had done a little bit of a background too, so we could kind of be a little more prepared. And what we came up with is that specificity of the markers. So a lot of cancer cell lines, tumors have very specific proteins that are associated to them. And so these become the way to actually mark them specifically. You can actually take multiple tumors and tease them out just based on this. I know we did a project with somebody doing breast cancer, and they were using the MUC1, and you can get very specific to those particular things. So I know in that paper, it was saying that some of it was dependent on if you were getting from the multivesicular bodies, if you were getting the exosomes, you had certain markers that worked very well. And on the opposite end, you had markers that didn't work there, but worked when they actually bled from the surface, more of in a microvesicle type portion. And just to elaborate a little bit more, it's the size. So basically based on the size. So yes, where they come from, ones that come from the multivesicular bodies, the exosomes tend to be somewhere between 60 and say 150. And then you get up into the microvesicles, which are more like 150 to 350. And those each had different labels that worked for them and didn't work for the other. So even within the same cell, you can kind of start to tease out based on the actual proteins associated with the surface. So that particular one, if you've done the preliminary where the cells themselves label specifically for that marker, then I do not see why it would not work very well for the extracellular vesicles.
Matt, will you share that paper with that, right?
Yeah, I just have the question from the attendee, but we can dig into that the link that you all shared back in our discussions yesterday. I think with that, since we don't have any more questions that have come in on the chat, and I think we've covered the biggest ones, at least specifically or generally that were submitted, I want to thank you both so very, very much for your time and energy in this. We wanted to have some fun, and I think we had some fun and a couple of smiles. And if you, for those of you that are in attendance, if you enjoyed this format, let us know. This is something that we're playing with a little bit, and we want to see if it's the kind of thing where we can all meet virtually and share our time and talent and experience with each other. So I thank everyone for making time in their day to join us. And with that, I will close it. Unless Alfonso, John, do you have any goodbyes or thank yous?
Yeah, thank you, Matt, and the rest of the team, especially with Lama and Martinez. Thank you very much for this opportunity. I always enjoy working with John. It's a big pleasure. We complement each other quite well as far as the BRLO does. But definitely it's a big pleasure. And thank you very much for this opportunity. And I hope that people have enjoyed this series.
Yeah, I mean, I would say the same thing. Thanks, to the people that helped put this together. This was supposed to be a course and a learning experience in Dublin for for those in the European Union there. And obviously things came up where we changed the format, but we wanted to give it a different feel. This was completely new to me. Those who know me know I have no problem talking and just this and chatting. So it's even easier because you just get into a conversation with your friend, right? And so you talk some science, you learn some stuff. It's like I do every day, going to my colleagues and dropping ideas around and trying to figure out what's the best move. So I thank everybody who watched it. I watched it and it was hard for me watching, even think of the content. I just kept saying, what was I doing? What was I thinking? Why did I do it? And then parts where I was like, I should have said that. Or no, that was a good kind of response. So also, if anybody has any questions, I feel free to, I don't know if you can reach out to Matt. I mean, I guess if we got a bunch of other questions, once more people watch the series, have no problem doing this again, kind of answering those questions or just doing it through the keyboard. So thanks, Matt. You did a great job behind the scenes and keeping us, I guess, on track, also keeping us focused. Because we digress. I do that a lot. Same thing. Thanks to Lama and Martin and Marcus for helping us put it together. Because otherwise, I'm not sure we would have even known where to start. I'm getting old. I got to ask my kids how this technology stuff works. So thanks to everybody again. Because without you, then it's just me talking to this screen. So.
Thank you guys both so much, and hope everyone has a wonderful day. Stay safe out there, and we look forward to doing this again sometime in the future. Have a great day. Thanks.
Thanks.